And there's 34 different linkage groups. Each of these corresponds to a chromosome in Chinook salmon. And each of these lines is an individual marker ordered along the linkage group. The pink, in this case, denotes the centromeres, since she was able to place those on her linkage groups. A lot of studies can't place the centromeres, but it is extra information if you can. So linkage maps are important because knowledge of the positional relationship between markers is important. You can imagine sort of a simple situation. You have a lot of loci that you've looked at. You've scanned for, or you've looked at FST across these loci and set a threshold, and you found, a number of loci that look like they might be adaptive. And at this point, it's interesting. There's a lot of loci that might be doing something, but if you can place those in order along the genome, you might see something like this that Hammer-Hansen found in Atlantic cod, which was 3 strong peaks of divergence between life histories of Atlantic cod. So there's 2 different life histories that interbreed. The rest of the genome is pretty much undifferentiated. But within these regions, they actually have inversions that prevent recombination between the 2 ecotypes. So because of the information that linkage maps give on marker order, they're really a foundation for a lot of different types of studies. QTL and GWAS studies look at genotypes across linkage groups to look for genomic regions associated with traits. Genome scans look for patterns of heterozygosity or FST or other population parameters across genomes. You can compare, or you can use linkage maps to identify genomic structures. You can identify regions of genome duplication. And you can compare linkage maps across populations or species to look for inversions or other chromosomal structural changes. So Larson et al. Did a genome scan where they were looking for differentiation between 2 life histories of sockeye salmon. So sockeye salmon in the Wood River have a pretty wide distribution throughout the system, and in general, population structure is correlated with geography. But you also have 2 distinct life history types there. There's a beach or a stream spawning fish, which is this really shallow-bodied one on the left, and beach spawning fish, which are really deep-bodied. So even though if you just look across the genome, there's not really much differentiation, they look very, very different. And in this case, it's sort of an obvious selected trait because the beach spawners, deep bodies are favored by sexual selection. And in stream spawners, the streams are so shallow that fish need to have a shallow body in order to navigate up the streams. So there's strong differential selection between them. So he made a linkage map and was able to identify this peak of differentiation between the populations. So you can see this peak marker has an FST of 0.3, which is huge on a background of no genomic differentiation. So what we did with this study is after we had this region we were interested in, we aligned the Atlantic salmon genome to it, identified a number of genes that were within this region, and these 2 peak markers are actually inside of a gene called TOLC4. So then we went back to a transcriptome study that I had done with rainbow trout, got the open reading frame for TOLC4, and this marker, the little purple triangle, actually is a non-synonymous SNP within this gene. So in this case, it's potentially the actual causal mutation governing life history, differences between these traits. And in this situation, it's useful for management because previously managers in Alaska have been able to look at the fish coming in, identify what population they're going to, and adjust catch limits. But now they can also identify which ecotype it is, and further refine their estimates of which fish can be caught and which ones should be protected. You can also look for genome duplication. Salmon have an ancestral whole genome duplication, and there's large portions of some of the chromosomes that are still retained as duplicates. So this was a map by Waples et al. Using chump salmon, and in this case, it was actually using haploid offspring, which we create in our lab to allow mapping paralogs, which I'll talk about a little bit later. But for this, all of the yellow dots are duplicate loci, and the blue dots are just regular diploid loci. And these lines show the links between chromosomes. So what's been recently found in Pacific salmon is every species that has been looked at has 8 pairs of chromosomes that still share duplication. And there's a lot of chromosome rearrangements between salmon, different fusion events and fission events, but these 8 pairs of duplicates have been retained in all of the Pacific salmon species so far. You can also identify inversions. This top example shows how you can identify inversions by blocks of linkage disequilibrium. So this shows a block of really high linkage disequilibrium along the chromosome. This is what a normal sort of chromosome looks like. And this was a study in monkey flowers by Haleski. And within this block of linkage disequilibrium, there were a number of genes that were associated with life history variation, whether the plants were annual or perennial. And so it's potentially a way, if you have an inversion that has these genes, it's potentially a way to keep co-adaptive gene complexes together. This study on the bottom shows another way of looking at inversions. Here you actually see reversals in marker order when comparing genomes and maps. And this is with stickleback, and the inversions in this case were correlated with population structure. So what I've been most excited about, and then what I've written my paper about, is using genomics, or using linkage maps to integrate different genomic studies. So if you think about it, most genomic studies for a species are sort of conducted independently. So one group over here does a QTL analysis, another does a population genomic study, another group might do a transcriptome study, and so there's all these disparate data sets out there, and linkage maps can provide a common framework for integrating those. So I took families from 2 different studies to make a new linkage map for Chinook salmon. We were able to place about 15,000 markers onto this map. And then because different salmon species at least share whole chromosome arms, or chromosome arm synteny, even with the rearrangements between them, we're able to align Atlantic salmon genome sequence to our map in order to provide annotation information. Because one of the limitations that we do have with linkage maps is that most of these markers we can't directly annotate. We don't know what gene they are, and we don't necessarily know what genes they're near. So placing the genome on top of the linkage map gives us that information. I also added transcriptome sequence from another Chinook salmon study by aligning directly to the genome and also to the linkage map to also help provide annotation information. And then what we're really interested in was aligning these resources from other studies, outlier loci from populations up in Alaska, regions of elevated population divergence, and then QTL for thermotolerance and basic or other body trails. And so in this case, we're able to look through all of these studies, identify regions that may be interesting. You know, here we have a QTL at the same region as elevated population divergence. But having the genome and the ESTs added to the linkage map, we're able to provide annotations. So by combining, well, and this is the whole map, but by combining these studies, we're able to get some new inferences. The 2 peak QTL in the thermotolerance study, one of them had a heat shock protein under it, and the other QTL had the inhibitor for that heat shock protein. So that's pretty good evidence. Those are both involved in thermotolerance in the species. We also found that within regions of elevated population differentiation in Chinook salmon, a lot of genes that were within those regions were involved in thermotolerance and stress response. And the region we were looking in was a pretty wide area of Alaska that has different thermal regimes. They could have different parasite communities, different flow rates. So there's all sorts of opportunities for different adaptations for different stressors within those populations. So next, I'll focus on how linkage maps are made. And I'll be going back to some basic biology just to try to get everyone onto the same page and try to provide things to be aware of for constructing linkage maps. So linkage maps, like I said, use recombination order or use recombination information to order markers on linkage group. And we get this recombination information by examining offspring within a family. And ultimately, the quality of the linkage map is going to be determined by the types of genetic markers that you use. Next generation sequencing will have many more markers than something like microsatellite studies. The type of mapping family, whether you're using diploids, haploids, backcross, recombinant inbred lines. Also the number of families that you have in a mapping study and the number of offspring per family. So recombination is measured in map units and more commonly called centimorgans. So this is measured as the distance between genes for which one product of meiosis out of 100 is recombinant. So basically, if there's a 1% recombination rate, this is one map unit or one centimorgan. So if you think about the maximum recombination rate for any two markers, it's 50%. That's the point at which it's just independent assortment. So the maximum length you can have between any two markers is 50 centimorgans. And again, this distance is revealed by recombination in families. So if you have a parental chromosome, so one of these chromosomes has a big A and a big B, the other has a little a and a little b, we can look at the offspring meioses to measure recombination. So meiosis happens, recombination happens, and we end up with 4 possible gamete types in this. We have the 2 parental gamete types, and then we have 2 gametes where there's been recombination between these chromosomes. So when we calculate the recombination frequency, what we do is we look at the number of offspring that has each of these genotype combinations. And to get the recombination frequency, we take the number of recombinants, so 19 plus 21, and divide by the total number of offspring, which is 1,000 in this case. So these 2 markers had 4% recombination rate, and so they're 4 centimorgans apart. So one of the ideas that is sort of key in a lot of the early mapping programs was the idea of phase. So in this example, the big A and the big B are on the same chromosome copy, so they're in the same phase. The little a and the little b are in the same chromosome copy, so they're also in the same phase. So some mapping programs, you need to know the phase of the markers, which allele was on which copy of the chromosome, in order to map accurately. And so that's one of the reasons that a lot of other studies used F2s or back crosses, recombinant inbred lines, because it was easier to determine the phase. There are ways around that, though, even with F1 families. So the way that we detect meioses is through informative marker segregation. And informative markers, essentially, in most cases, are markers that are heterozygous in one parent and homozygous in the other. So if we go back to that example, we have our female parent. These are the 4 different meiosis products that you can have. And if we have a male parent, which is homozygous for both of these alleles, we can tell which of the offspring genotypes are recombinant. Either the little a, big B, little b, or the little a, or big A, little b, little b, are the 2 recombinants. So these 2 meiosis products are recombinants. What's important is that if you have both parents being heterozygous for that locus, then you don't really know which parental chromosome it came from. So if both of these parents are heterozygous, have the same meiosis products, there's quite a few different offspring genotypes, but when we look at the offspring, we don't know which of the B's came from which chromosome, which of the A's came from which chromosome. And so generally, these aren't able to be mapped, but reality is more complex. So if you look at this figure, this is all of the possible informative marker types, or all the possible marker segregations for a marker that has 4 alleles, or up to 4 alleles. So if you have 2 parents, each has 2 chromosomes, you can have 4 alleles, A, B, C, and D, and then O are no alleles. The key thing to get from this is any of these that say asymmetrical, those can all be mapped. So these are all the different segregation types that can be put onto a linkage map. These that are the ones that are asymmetrical. So these that are symmetrical, the hybrid by hybrid, and then homozygous for both. Those are markers that in most cases can't be mapped, but sometimes they can. If we know phase information, then those can be mapped. But in general, these markers aren't very reliable for linkage maps. So we tend to exclude them. And this has actually really important implications for linkage maps, because if we do a normal mapping family, you just have a parent or a female parent, male parent, you look at the offspring. When we do the mapping, we look at loci that are heterozygous only in the female and heterozygous only in the male. And so when we do that, we end up with a female map, and that's all the loci that are heterozygous in the female, and we end up with a male map, which is all the loci that are heterozygous in the male. And so with a single family, your two maps won't actually share any loci. And then these maps also ultimately measure recombination in each individual sex, which can be very different. And I'll show some examples of that. So to assemble linkage groups, markers are grouped together also by recombination. Alleles for markers on the same chromosome should be inherited together, more often than expected by genes. Markers on different chromosomes should be inherited independently. So the A and the B loci in this case should tend to have alleles inherited together. The C and the D should have alleles inherited together. And the A and B genotype should have no relation to the C and D genotypes. So the likelihood that markers are inherited together are often given as a log score. And this is just, and then likelihood thresholds that use this log score are used to group markers together. So one of the consequences of this is if you have a loose threshold, you can have unrelated linkage groups merged together. If you just have a few loci that by chance have similar inheritance patterns, then linkage groups can collapse into a single linkage group. And then if you use thresholds that are too stringent, you can actually end up fragmenting linkage groups and artificially splitting them. So this is how to calculate the log score. I wouldn't bother too much with this. Most programs do it automatically for you. But if you want the information, it's there. So this is an example of how varying these thresholds can change the amount of linkage groups that you get. So with Chinook salmon, 34 linkage groups is the correct number of linkage groups. There's 34 chromosomes, we should have 34 linkage groups. And in this data set, it's actually pretty robust. Varying the log threshold anywhere from 14 to 22, we get the right number of linkage groups. But if we go below 14, then we start combining linkage groups together. At the extreme end, if you use a log threshold of 1, every linkage group gets merged together. And then as we increase the log threshold up above 22, then linkage groups start splitting apart. In my experience with Chinook salmon, they usually tend to split apart at the centromeres for metacentric chromosomes. Those are what tend to split apart first. So once you have your linkage groups, or once you have markers combined into your linkage groups, then they're ordered within linkage groups. And so if you have a simple case with 3 different markers in a perfect world, you can measure the centimorgan distance between each of those marker pairs, and then you can use that to identify the order of the 3 markers. So it's conceptually pretty easy to do, but in reality, it's really complex. So this is a figure from Wu et al. Looking at how to combine multiple families for a linkage map. So it's a little hard to read, but markers aren't all shared between the families. And then sometimes there's differences in marker order. So in this case, this family has marker 4 up here marker 4 is down here. And so this can happen because of genotype errors. It can happen because segregation isn't always perfect. Sometimes one locus gets inherited more than another in different families. And so it really can be computationally difficult to try to order maps either within families and especially across families. So there's a few different expectations that we have for linkage maps that can help us to build them. One is that linkage groups should correspond to chromosomes. So we should have ideally 1 linkage group for each chromosome. Linkage group size is actually dependent upon recombination. So it's not necessarily related to physical size of the chromosomes. And recombination rates vary between species, between sexes, between chromosomes, and even along chromosomes. Recombination is not physical distance, which is a very important point to make. And they can be very, very different. Interference, which Fred talked about a little bit on Monday, will influence linkage group size. So if you have, so interference limits the number of recombination events per chromosome. So in a species with complete interference, you only have one recombination event per chromosome, and you end up with a maximum of 50 centimorgans per chromosome arm. So an acrocentric chromosome should be about 50 centimorgans long, and a metacentric chromosome should be about 100 centimorgans. And in salmon species, there is near complete interference. So this is a good guideline for us to use to see if our linkage groups are looking correct or not. And interference varies heavily by species. So this is an example of the Chinook salmon map that I made on the left, and then a barley linkage map on the right. And you can see all of these chromosomes here are metacentric chromosomes, and they're near 100 centimorgans long. Most are a bit longer. These are all acrocentric. They're close to 50 centimorgans. In this barley map, one obvious thing is there's many fewer chromosomes. There's only 7, but also the longest are near 250 centimorgans. So there's much less interference in this species. This is an example of a sex-specific difference. So in Atlantic salmon, a female linkage map is on the left and a male linkage map is on the right. And you can see for some of these linkage groups, the actual distance is pretty similar. Chromosome 1 is actually pretty similar between the two linkage, or between the two maps. But there's a really striking difference in the distribution of markers along the linkage map. And that's because of how recombination takes place differentially in male and female salmon. So this plot here shows the male centimorgan position on the bottom, or on the x-axis, and the female on the y. So you can see this whole 100 centimorgans of the female linkage map is within just a couple centimorgans of the male map, right in the middle. And the male map, the entire 100 centimorgans, is just in these few centimorgans at the very end. So in male salmon, recombination takes place heavily at the telomeres. And in female salmon, recombination is more towards the centromeres. And so there's very large differences in recombination position. So for salmon, because of this, and you can easily see there's not hardly any real distance information, or real relationship on information on marker order in the male map. There's much more information on marker order in the female map. So for salmon, most people just generate female maps and don't even bother with male maps. They're also much harder to assemble in salmon. In some species, you see the opposite. Some species, female map, or some species, the male maps are desirable. But it's pretty common for recombination rates to vary across sexes. So for mapping studies, when we think about what type of mapping family to make, in our lab, the first consideration is do we want a haploid family or a diploid family? We can produce haploid families in our species, and it's a pretty straightforward process. And there's some advantages to haploids. One of them is that haploids, so go to the next slide. So diploids are the norm for most species of interest. But for haploids, they naturally occur in some species. They can be produced in others. And the advantages are that with haploids, you have a lot less genotype error. In the absence of a duplicated genome, you just expect that all loci are heterozygous. And so, if with that expectation, there's not going to be much genotyping error there at all, or they'll all be homozygous. Because of this, you can map more loci. If you remember what we were talking about earlier, loci that are heterozygous in both parents generally can't be mapped. If you have a haploid family, any locus that was heterozygous in the parent of that family can be mapped. And so we're able to map more markers. We can also map duplicate regions, which is important in salmon, not necessarily for other species. And we can also use haploids to place centromeres on linkage maps. So this is an example showing the difference between a female or a haploid and a diploid linkage map in Chinook salmon. And in this case, the diploid or the haploid parent was the same female parent of the diploid map. So the genotype was the exact same in the parent. We were able to map 5,400 loci in the haploid map. For the diploid map, we were only able to map 3,700. So we lost a lot of loci in the diploid map because of a combination of heterozygosity in the male parent and then not being able to map duplicate regions in salmon. The male map did have 3,500 loci for the diploid family, and of course there's no male map for the haploid family. So there is extra information for getting the male map, but in salmon, because male maps are difficult to produce and they tend to be unreliable, we actually couldn't get a successful map out of that, so it wasn't useful information anyway. If you do haploid families, you can also use them to replace or to place centromeres by plotting the recombination fraction. So this is an example of a metacentric chromosome, and when you plot the recombination fraction, you just plot it from either end of the linkage group, and for a metacentric, where they meet in the middle is where the centromere is located. So the other advantage of haploids is mapping paralogs. So paralogs are difficult to genotype and map in diploids. And if you think about it, there's 5 different genotype classes for a locus that has 2 copies. You can be completely homozygous, and then there's 3 classes of heterozygotes with different numbers of each allele. And in the old days with allozyme data, you could actually look at these and tell them apart. But with next generation sequencing, all of those just look like a heterozygote. And so we don't know what the true underlying genotype is, and we can't map it. But with haploid individual, and then with salmon, these also have a strong positional bias. They tend to be in the telomeres, so we lose a lot of information by not including these. So with haploid mapping, essentially, if you have your duplicate chromosomes in a diploid, in the haploid offspring, they've separated, and these essentially act like normal diploid loci. So they're really easy to map with haploids. So the way that we produce haploids is through irradiation of the sperm or the egg. So we basically knock out the DNA in one of them. We usually knock out the DNA in the sperm, and then the sperm fertilizes the egg. You have 1 copy of the DNA, and then we sample them at hatch for DNA. Irradiation of sperm or egg is really common in salmonids. It's also been used in a lot of plant species. There's also species that naturally produce haploid offspring. In some plant species, the megagametophyte is haploid, and so you can just sample that and use that for your mapping. Just go collect a bunch of seeds and you have a mapping family. And finally, newer technologies are allowing sequencing of single cell recombinants. So just direct sequencing of sperm or eggs. So in this case, they isolate DNA from a single cell, amplify it, and then sequence it. And so you don't even need to produce a mapping family, you just need samples of sperm or eggs. And this actually has some real benefits for a lot of species. Linkage mapping usually requires a lot of offspring to get reliable maps. And in some species, large families are just impossible to produce. Either because they just don't have many offspring, or generation times are really long. So you can sequence egg or sperm directly in these species to make your linkage maps. But another thing to be aware of is how recombination occurs in each sex. Is it worth sampling the sperm from a species that isn't going to yield a good map because of male recombination? One example is female Lepidoptera. You could go sample eggs from them to make a linkage map, but there's absolutely no recombination in female Lepidoptera, so you wouldn't be able to do anything with it. For anyone that's interested, there's a review on single cell sequencing by Rawad et al., 2016, and I'll have the references to for anyone that's interested. So for sort of traditional mapping families, for regular diploid individuals, single generation crosses, such as F1, is just a normal cross between a male and a female parent. And these used to be relatively uncommon, but with some of the new mapping programs out, they're actually really easy to do. And we don't have some of the computational issues we used to have. A lot of the older studies had multi-generational crosses, F2s, back crosses, recombinant inbred lines. And these were really useful when mapping programs had to know the phase of markers, because you can infer the phase from multiple generations. So one of the trade-offs with mapping that you have to make is how many families are you going to have and how many offspring are you going to have. If you assume that there's a limited amount of sequencing that you can do, there's just a choice that has to be made. And there's a fundamental trade-off associated with this. If you have more families, you have more loci that are heterozygous in the different parents, and you have more markers on your maps. But if you have more individuals within a family, you have more recombination events, and you can get greater resolution. So most of the time, people are interested in the number of markers that are on the map. And getting the most markers on the map is the priority. But sometimes resolution is beneficial. If you're doing a genome assembly, it can help to have fine-scale resolution between markers to help order chromosomes. And having fine-scale resolution can also help in QTL or GWAS studies for finer mapping of traits. So this is an example also from my Chinook map showing the effect of having more families. So in this case, each of the 4 families had around 5,000 or 6,000 loci within each individual family that were informative and able to be mapped. When you compare the families, each pairwise comparison only shared about 25% of the loci. And what was really striking is there's only 261 loci that were informative in all 4 families. As you combine more families, you get more loci on the map, but there is a diminishing return. So with our first family, we had 5,500 markers mapped to the map. It's the first family. We had our second family, there's only 3,400 additional markers. Our third has 2,800, and our fourth has about 2,000. So as you add more and more families, there's less of a return. And then one way that you can actually get around this is try to add families from divergent populations. So since this, we've taken 4 more families from Alaska. And within the Alaska populations, we see the same thing. And we end up with a map of about 14,000 markers in Alaska. But when we combine the 2 maps, we get a map of 24,000 markers. So there was a lot less overlap between Washington and Alaska than there were between the Washington families. So if you do want to increase resolution, you need to increase the number of samples per family. So resolution on average should be 1 Morgan over the number of samples. So if you have 100 samples on average, you should be able to get 1 centimorgan resolution. So for my linkage map, we had 545 samples. So you would think naively that the resolution is 1 over 545. So it's 0.18 centimorgan resolution, which is pretty good. But what we actually observed was the resolution was 0.68 centimorgans. And the reason was most markers are only genotyped in 1 or 2 families. So if we look at the average number of markers, or number of individuals genotyped for a marker, what we found was that the average was 154. 1 over 154 is 0.65 centimorgans, which is really close to what we actually observed. So if you want to build a linkage map, in summary, you really want to use the expectations of the organism to help you decide if your map is of good quality or not. How many chromosomes are in your species? Are you finding that number of chromosomes in your linkage map? If you have too many, it could be fragmentation. If there's too few, it could be over-merging. How long should the linkage groups be in your study species? Do you know anything about interference in your study species? Is there any evidence from other studies showing how long linkage groups should be? Are there sex-specific differences in combination? It's really common, but some species males have more, some species males have less. It can really vary a lot. And the data quality will really affect the results that you get in the linkage map as well. Better data makes better maps, and better data is always better. But filtering in linkage maps is actually pretty straightforward because you basically have an issue of missing data and wrong data. So you have miscalled genotypes and the absence of genotypes. So for samples, I usually filter on genotype rate, and our genotype rate is usually very high in marking families. We have over 90% genotype rates usually. We also look at heterozygosity, and we just plot heterozygosity across all of the samples and look for individuals that have unusually low heterozygosity or unusually high heterozygosity. Usually what we'll see is if a sample has a low heterozygosity, it also has a low genotype rate, and that was just because it had a low sequence amount for that individual. For filtering loci, we just filter on genotype rate, and then we can also filter on segregation distortion. Does the locus fit expected patterns of Mendelian inheritance? And segregation distortion is interesting because there can be real biological reasons that markers don't get inherited according to Mendelian expectations. And so I'll actually show an example of, in the practice or in the hands-on data, there's instructions to filter loci by segregation distortion and to just go ahead and make a map of that or ignoring that filtering. And you actually get a very different result because of, we think because of the genome duplication in some lines and how some chromosomes can get inherited. So one of the common issues with linkage maps is fragmentation or overmerging. And these can actually be really difficult to identify in the absence of something to compare to. If you don't have a genome or another linkage map, it can be hard to tell if there's fragmentation or overmerging going on, because you could have a situation where you have the correct number of linkage groups, but one of those linkage groups is a combination of two, and another linkage group is fragmentation, or another two are fragmentation. So you have the correct number, but the linkage groups are still incorrect. In this case, this shows overmerging of a linkage group in salmon, and you can see it's obviously very different from everything else. So a good thing to do is just compare all your chromosomes, see what they look like. Particularly with what happens often with overmerging is you'll have a few loci that had similar segregation patterns between the two chromosomes, but nothing else does. And so it has a lot of issues ordering it, and it'll end up with large gaps or extremely long distances. And so overmerging often can be pretty easy to detect because they'll end up looking like this, just massively inflated linkage groups. Fragmentation can be a lot harder to detect unless you know how many linkage groups that you have, or supposed to have. If you can't get a setting for your linkage map that gets the correct number of linkage groups, if you always either have overmerging or fragmentation, choose fragmentation. Overmerging introduces all sorts of issues with order because it's trying to merge unrelated linkage groups. If a linkage group is fragmented, the order within each of those fragments should still be correct. They're not joined together correctly as they should be, but all of the relative positions within each of those fragments is still right. Gaps are another thing that we see pretty often. These can be signs of genotype errors. It can be incorrectly placed markers. The program didn't put it in the right spot. It could also be a sign of overmerging, like we saw on the previous slide. In this case, this linkage group had a really long gap of about 40 centimorgans, with just a couple markers out there. In this case, it was pretty easy to do, pretty easy to fix. We just trimmed off those markers. We think they really belong to that linkage group, but the distance obviously is not correct, and it was only 3 markers, so we didn't really care about those. In other cases, it can be a little harder. If you have a large gap right in the middle, then it's not really clear, how to fix it. Then you can start going through markers one by one, seeing if any of those have signs of segregation distortion. Is there any evidence of incorrect genotypes? And sometimes it may be that you just end up splitting the linkage group in half and treating each half separately. So another issue can be unrealistic distances. So if you have expectations of how long your linkage group should be based on biology, how do the lengths compare? In this case, the linkage group the linkage map on the left is a Chinook map that doesn't have duplicate loci on it. This one on the right does have duplicates, which are all pink. And the one on the left is essentially missing some of these large regions of the genome. And so we have linkage groups that are only 15 centimorgans long, where they should be about 50. And so this is an example of a map that we know is too short, but we also have a reason that it's too short, because it's missing whole regions of the genome. This is an example of a map that's too long. So this is the exact same data, but two different mapping programs that we used. The one on the left, we have linkage groups that are over 300 centimorgans. It's just way too long. So that was one that I made early on when I was doing linkage mapping. And what we used to do is we would make a map for each individual family. So we had 4 different families for this. We made a map for each family individually, and then we combined those with a program called merge map. And what happens is, when we look at each of the individual families, the distance is matched up with this. And it matched up with our expectations. 50 centimorgans for acrocentric linkage groups, 100 for metacentric. But when we started combining families, the distances kept getting longer and longer and longer. So we tried different combinations of families, added in different orders, but basically every family that was added increased the length 30%. And what happened is merge map, when it saw conflicts in marker distance, generally took the longer distance. And so it just ended up with this artificial inflation of the map. And some of the previous studies that had come out that had seen really long map lengths in salmon had used the argument that as you increase the number of markers, you increase the size of the linkage group. And that's only true if you're missing parts of the linkage group. In this case, it was just an artifact of the program. The map on the right was produced with a program called LetMap, which can take into account all of the families at the same time and look at recombination events within families, look across families for markers, and gets a much better map. In this case, it fit all of our expectations for lengths. On the plus side, even though the distances were completely wrong on the map on the left, it's still actually the relative order of the markers was pretty good. If you compare the order on the left to the order on the right, it's a nice plot. There's false fine scale resolution on this. There's markers that are separated that really shouldn't be, but the relative order of everything was still correct. So if you have maps and you compare across populations or individuals, then you can start making some nice inferences. So Larson et al., as part of that study I talked about earlier, did a linkage map for sockeye salmon. And by comparing male and female sockeye salmons, he was able to identify karyotype differences between them. So male sockeye salmon have a single metacentric sex chromosome, and female sockeye salmon is broken up into 2 acrocentric chromosomes. And we can see that by looking at sex differences. In some cases, this wouldn't necessarily be informative. So you could easily imagine that you do a map for a male and a female, and one of your linkage groups is fragmented in one of your sexes. So it may not be a real biological reality, but it may have just been that the program couldn't merge it together. In this case, because of karyotype data, we were expecting that we would have a difference between males and females. And so this fit with what had previously been observed. So we were more confident about it. You can also look across populations or species to identify inversions. This is showing an inversion between allopolyploid strawberry, between different cultivars. How I often plot linkage maps is I'll do a dot plot of distances along 2 axes. And in that case, this is what an inversion looks like, where it switches the diagonal, and you have the markers reversed. So another thing that you can see is the movement of loci across linkage groups. And in a lot of times that would be a red flag. If you see a marker that's moving around, especially between linkage groups, that normally doesn't happen. And so a lot of times you see that and you think something's just wrong with your data. But in this case, the sex-determining gene in Atlantic salmon has actually moved around between different populations. So by comparing different populations, they were able to find that the sex-determining gene has moved across 3 different chromosomes across the species range. So if you see something like this, you really want to be sure about it before publishing it. And they did a lot of work to verify that that was a real result. So there's a lot of programs that you can use for linkage mapping. I've listed some of the programs that people still use these days. Some of the older programs that people used to use just can't handle the number of markers that are available now, or they can't handle like family sizes. So in these, the programs that I've used are MSTmap and MergeMap, and then LetMap, and I've played around a little bit with one map. They each have different advantages. MSTmap works on haploid individuals, and it works well, but your data has to be phased. So you need to know which allele came from which chromosome in the parent. And there's ways to actually determine that pretty easily that we were able to figure out. But if you don't know the phase, it reconstructs two linkage groups for every actual linkage group. It gives you one in each parental phase. MergeMap used to be the only way to really combine maps across multiple families, which is useful information to know the relative marker position, but it really inflates map distances. So I wouldn't recommend using it anymore because we have things like LetMap, which can combine multiple families really easily, works with thousands of markers, hundreds of individuals without a problem. The problem with LetMap is the documentation is really pretty awful for it. But the author is pretty responsive. So if you have questions, you can email him, he'll get back to you and help you out. One of the other things is LetMap is limited in the cross types that it can do. It's really only meant to do an F1 family. But there's tricks you can do to code the data in ways that other families can be used. We use LetMap with haploids, which shouldn't be able to be used with it. But essentially, instead of having an A and a B allele, you just call one a heterozygote and one a homozygote. And it works just fine. OneMap, JoinMap, and RQTL all handle a lot of different cross types. So if you're doing some other family type, it's a really useful, there's some really useful programs. But out of those, OneMap only handles a single family at a time. JoinMap can do multiple families, but it costs money. And I don't know how much money it costs. I've heard it's a lot, but I've never looked into it. It's also limited to only about 3,000 markers. In our current families with RADseq data, each individual family has about twice that. So we're already past the limits of JoinMap. RQTL has high memory usage, and the data has to be phased for outbred families, so it's more complex. The nice thing about LetMap and MSTmap is they both have relatively low memory usage. I can run them on my personal laptop, and it's not an issue. They're both processor limited, so it just takes more time to finish if you're running on a slower computer or one with less capability. So I guess it is time for the practical session. Before we get into it, I'm just going to go through the programs a little bit. There's going to be two data sets there. There's a haploid data set and a diploid data set. I recommend starting with the diploid data set in LetMap because it's going to run the fastest. If you wanted to do the entire linkage map, you could get it done within an hour. But you can at least get, so when you order chromosomes in LetMap, the shortest chromosomes go the fastest. So I have you start off with the shortest chromosomes, and you should be able to get a chromosome order within a few minutes for the short chromosomes for LetMap. So with LetMap, there's a few different modules for it. There's the filtering module, which allows you to filter markers for segregation distortion, missing data, presence in families. So you can, if you have multiple families, you can say you want this marker to be present in multiple different families or informative in those families. And you can also filter individuals for missing data. Then the other three modules are really where the work gets done. Separate chromosomes is what forms the initial linkage groups. It does this based on log thresholds, and it's very fast. So you can you put in your parameters, choose a log threshold, run it, and within a few seconds, it'll have grouped your markers into linkage groups, and it will tell you how many linkage groups you have. I have some summary scripts in the handout that you can use to see how many markers are in linkage groups, how many linkage groups there are. And you can vary the log threshold, change parameters, and you get a pretty good idea of how the linkage map is behaving. So when you run it, separate chromosomes is what you use to determine how many linkage groups you have. So that's the first step. Join singles attempts to add the remaining markers to those linkage groups, but using lower log thresholds. And then finally, order markers is what actually orders the markers in each linkage group. And there's a description of each of these and how to run them in the handout. The input format for LetMap is pretty simple. It's called a linkage format, and I should have a link on the handout to the description of that format. So the first column, which is in the blue here, is the family ID. So if you have multiple families, you just list them sequentially. Then you have your sample. You list the sire and the dam first for each family. And then you have the sire of each cross. If it's the parents, they don't have a sire, so it's 0. The dam for each cross, and the same thing. If it's the parents, you just put in a 0. You put the sex for each sample. This is really only necessary for the parents. If you know the offspring sex, you can put it in. If you don't know it, just put a 0. The next column is a holdover, because this was used for a different mapping program previously. They used to put in covariates such as different traits or morphologies to try to see where these were relative to markers. So I just always code that as 0. And then finally, you have your genotypes for each of your loci. There's multiple ways that it can accept genotype coding. I've just coded it as a simple 1-2 system. 0s are missing data, 1-2 is a heterozygote, and then 2-2s and 1-1s are homozygotes. So one of the things to really be aware of when you run LetMap is that when you get the output, it'll have all your markers, all the orders listed for them, but the marker names are not the marker names you put in here. So the marker names that you get out of it are the order of the markers in here. So marker 1 on the linkage group is this. This is marker 2 and marker 3, and so on. And so it was really confusing the first time I tried to run it. I was trying to figure out what my locus names were. So what I always do is this first line, it's not actually necessary in the input file. You don't have to have it there. But I always have this first line, and then I just copy it out, I transpose it into Excel, and then I just autofill down to get the order for each of the loci. So it makes a nice lookup table. This is locus 1 in the output. That's locus 2, locus 3, and so on. And so it's a nice way to ensure that you're getting the correct names when you have the actual output. So if you want to get the full commands for each module of LetMap, you can just type in the command to run the module itself without any settings, any options. So LetMap runs through Java. It doesn't install like other sort of programs. You can have it anywhere on your computer. You just need to call Java and then tell it where to look. So the way that it is set up in the virtual or in the Docker is if you type this in anywhere on the machine, it knows where to look for the LetMap program, and this will give you the settings for separate chromosomes. And the commands that I've included in the handout are all written like this. You should be able to run them from anywhere in that machine. If you want to run it, if you wanted to take this and run it on your own computer, or you wanted to run it somewhere else, you would have to change this part of the path, the bin code in bin. You would just have to look and see what's the file path for the separate chromosomes folder, or the separate chromosomes program in the folder. So there's a few specific commands that I have you look at in the handout. One of them is informative mask. This tells it whether to look for loci that were informative in the male parent. So these were heterozygous in the male parent and homozygous in the female. You set that with informative mask equals 1. If you want to look at loci that were heterozygous in the female parent, then informative mask is 2. If you want to have loci that were heterozygous in both parents, then you set it to 3. And you can also just leave it off, and it will look at all loci. So certainly play around with that and see what happens. You set informative mask for each of the different modules in LetMap. So just keep it consistent as you're going through. LOD limit sets the threshold for aligning the markers to linkage groups. So you'll use this in separate chromosomes to group your markers together in the linkage groups, and then you'll also set it in join singles in order to join the additional markers. So play with that and see how many linkage groups you get out of it. Size limit sets the minimum size to include linkage groups in the output. So I've set it at 10, so any linkage group smaller than 10 markers isn't going to be output. When you run it, it'll show you how many linkage groups are discarded because they're too small. But you can try varying that around and see how many linkage groups you get out of it. And then I also have a haploid data set that you can run with MSTmap. And it turns out, well, it's a little more complicated with MSTmap because the input file I have in there now is meant to run through a whole pipeline for identifying paralogs and splitting it out, and then mapping the paralogs afterwards. So it's not formatted for MSTmap yet. So I will, if nothing else, I can just email you guys the correctly formatted file to run that. One of the important differences between MSTmap and LetMap is it's made to work on haploids. So in this case, individual alleles are coded as A or B, and you don't have heterozygous loci. A and B are assumed to be different chromosomal phases in the parent. If you don't know the phase of the markers, like we don't at this point, then you end up with 2 linkage groups for every real linkage group, one in each phase. And I'll show a slide next that shows what that looks like. We can, if we had a multi-generational study, we can infer the phase, but we actually do something called bulk phasing, which is really easy to merge the linkage groups. So if we imagine a case where we have all of our markers that really belong in a linkage group, at this point we sort of arbitrarily assigned an A and a B to each allele because we didn't know what the parental phase was. So if you just looked at the A's and B's, along the chromosome, this is what it would look like. When MSTmap goes to actually map it, you end up with 2 linkage groups, one in the A phase, one in the B phase. And so we want to be able to combine these 2 linkage groups correctly into one linkage group, because each of these linkage groups has about half of the markers that it should have. So when we do bulk phasing, we just take this data, we duplicate it, and then we switch them all to B's. We take this data, we duplicate it, and we switch it all to A's. And I have a script in there that will do it automatically. But once we run that, what we end up with is 2 chromosomes, or 2 linkage groups, with every marker on it in opposite phases. And so from there, we just get rid of one of these. I have some scripts to match up chromosomes and identify them based on shared locus names. And then what's nice is once we have this data, it's also been phased. And so if you want to do something like centroid replacement or look for recombination events across chromosomes, you can identify that. Because as you look across an individual, if you just place all of its genotypes with the phase data on the linkage map, a recombination event is where it switches from A genotypes to B genotypes. So you can just look at the data and see where recombinations are. So this is an old plot I had when we were first testing out the bulk phasing method and what it ended up with for the MSTmap output. And you can see basically each real linkage group has 2 sets of chromosomes that it assembled. And then from here, I just took out one of them for the publication because they're just duplicate linkage groups. So the format for MSTmap, all of this information at the top is the instructions to run it. So everything's in one file. You don't give any commands in the command line of the program. You just give it the input file, and all the commands are in the input file. And then after that, you have a column of locus names, and then individuals, and your genotypes for each individual as either A or B. The parameters that it has are population type. We tell it that ours is double haploid. If you have other cross types, you can use those. The population name parameter is the population name, call it whatever you want. The distance function is important, whether it's a Kasambi or Haldane distance function. And Kasambi assumes that there's no complete interference, So that's appropriate for our species. The cutoff p-value is actually the threshold for combining markers to linkage groups. So it doesn't use a log threshold like LetMap used. It has a p-value for whether markers are inherited together or not. And what's interesting is if you go to the online version of MSTmap, because you can run it through a website, they actually have a lot score now. They don't use this p-value cutoff anymore, but it's still in the command line version. The no-map distance is an interesting, well, the no-map distance and the no-map size are an interesting way of trying to get rid of bad markers. So you actually have, it uses a combination of these two parameters to determine whether to get rid of markers that are at the end of a linkage group. So basically what it says is it's going to cut off markers at the end of the linkage group if there's fewer than a certain number of markers and they're more than a certain distance away. So if you have no map distance set to 10 and no map size set to 3, if you have two markers that are 11 centimorgans away from the rest of the linkage group, it'll cut it off. If you have 5 markers that are 11 centimorgans away, it'll keep them. So it's just trying to trim off spurious markers at the end, but it's a sort of weird way of doing it. The missing threshold is the proportion of missing data that is allowed. In this case, I think all the data has been pre-filtered anyway, so I don't think that's actually going to take out any markers. Estimation before clustering tries to estimate missing data before combining markers to linkage groups. Detect bad data tries to detect problem genotypes, and it just prints out a list of loci that it thinks are an issue. And then the objective function is just whatever method you would choose to tell it to sum the combination events. And then the number of loci, number of individuals are the number of loci and samples in your input files. The full parameter descriptions are available there, and I think that is in the handout as well. So if you want to run MSTmap, you basically just follow this example in the directory where MSTmap is. And the file, it's mstmap.exe, then the input file and the output file. The command should be in the handout. And the MSTmap program actually will probably take about an hour or so to run, which isn't bad, if you're trying to make a linkage map, but it's a little long for this. And I forgot to include an example of the output file, so I'll be sending that around as well, if you want to look at those. And that is it. If there's any questions, I'm happy to take them. Otherwise, we'll run the handouts. And I will be sending out the correct data for MSTmap.